one can use the definitive zero is wrong. However, one can make sure that this quantity here, that is the other information, goes to infinity as we move in space. We're going to make sure that this function here goes to infinity. And of course, this provides an exponential suppression. And so even if there might be boundary terms that are exponentially suppressed, and so we're sure that is that clear? That the QV potential is positive, right?
It's a multicellular animal that contains billions of cells, each with their own intrinsic properties. And so our bodies contain many different kinds of cells, and these cells often have to move some distance from where they're born to where they need to end up in order to carry out their functions in the animal. So in my lab, we're interested in understanding during development and how many cells work together to build functional three-dimensional structures. One way to think about this topic is imagine a crowd of people at a busy train station. Each person has somewhere where they're going, but how they get there can be quite complicated. So to get where you're going and navigate through this complex environment, you need to know where you are in space on multiple levels. For example, you need to know where you are relative to the 4 or 5 closest people around you, but you also need to take into account the overall flow of the crowd, the push and pull of crowds of people as they move to the station, as well as know where you are on a map relative to your final destination. And all 3 levels of information are necessary to take the most efficient path to where you're going. So in my lab, we're interested in understanding how cells navigate as they move through the body during development. And to get to the places where they need to end up, cells receive spatial information from many different sources. So for example, cells can interact with other cells in their immediate environment. Cells generate forces in order to move, and they in turn are exposed to the forces generated by cells around them. And finally, each cell needs the equivalent of a compass that tells it where it is relative to a global map of the entire animal. Shown here, a map of the Drosophila embryo, fruit fly embryo. So all 3 types of information are necessary to build the elaborate structures that are generated during development, as well as to enable the regeneration and the repair of these structures in adult animals. So today I'm going to talk about an organizational problem faced by many animals, which is how to build a body axis that's elongated from head to tail. And this is a process that happens in fish, it happens in frogs, and it happens to you, or you wouldn't be watching this talk. And during this process, all of these animals need to reorganize an amorphous mass of cells that can be hundreds of cells or more inside into one that's highly elongated along one axis to produce the characteristic elongated shape of the body plan. Now, in my lab, we study this problem in the fruit fly Drosophila, shown here on a banana. And Drosophila is an excellent system for studying developmental biology. The flies are very easy to grow and work with in the lab. And importantly, the structural changes that build the head-to-tail body axis happen at the surface of the embryo, where we can directly visualize what the cells are doing by live imaging. Now, it's important to understand this process, not only because many different types of cells, different types of tissues have to elongate during development, such as the lung, the gut, and the spinal cord, but also if we can understand how cells self-organize to form complex structures during development, we would be that much closer to helping to fix structural defects that arise during human disease. So this is a picture of the Drosophila embryo undergoing elongation, and the embryo before elongation is shown here at the top. It's about 0.5 millimeter long, and at this stage, the embryo is basically just a single monolayer of cells surrounding a ball of yolk. So all the cells are located at the surface of the embryo. And the head is to the left, the tail is to the right, and as during elongation, the germ band indicated by the yellow line undergoes a major reorganization in less than 1 hour to more than double in length along the head-to-tail axis, and at the same time it shrinks to half its initial height along the perpendicular axis. And because the embryo is encased inside a protective membrane that surrounds the embryo, as it gets longer, it's forced to wrap around on itself so that the head ends up next to the tail. Now, Ken Irvine and Eric Gustav showed that these structural changes occur primarily through organized cell movement. So while the embryo as a whole is elongating from left to right in these images, individual cells within the embryo are moving up and down in a coordinated fashion, and this is shown in these the pictures here. And what you can see is that cells that basically rows of cells intercalate to form fewer longer rows. So for example, if you look at the cells in this yellow row here, by the end of the process, the yellow cells that were all originally next to each other in a row become separated by neighboring cells from the green and purple rows. So to understand how cells reorganize to get from here to here, Gene Farrell and Ori Weiss in the lab came up with a computer algorithm that can automatically identify and track cells in time lapse movies. And what they found is that individual cells not only navigate as solitary cells through the tissue, but they actually engage in collective cell behaviors that enhance the speed and the efficiency of elongation. So this slide shows the 2 types of cell rearrangements that contribute to elongation. Each row of cells shows the same group of cells from 3 time points from a time-lapse movie. And the top panels show the process of local neighbor exchange, discovered by Slomalik, Lee, and colleagues, where a single vertical edge between 2 cells, shown in red, shrinks to a point and is replaced by a horizontal contact, here in yellow, between the top and bottom cells that were previously not in contact with each other. In addition, through live imaging of many cells, we were able to find that these cells also engage in this unexpected collective rosette process, where many linked edges, shown here in red, shrink simultaneously, leading to the formation of a multicellular rosette structure where as many as 10 cells can come into contact at a single point. And these structures always form and resolve in a strictly directional fashion, so they form through the contraction of vertical edges, and they resolve through the formation of these new horizontal edges, shown here in yellow. And what this does is convert a group of cells that was initially tall and thin into one that's now short and wide. And you can imagine many cells are doing this throughout the tissue, that this would cause an overall elongation of the embryo. So to show you what this looks like, this is a movie of axis elongation in a trophil embryo where each of these polygons is a cell, and the cells are outlined by the green fluorescent protein tagged to a membrane marker. And the head is to the left, the tail is to the right, and the cells that we're interested in are in the center. And as the embryo elongates, the cells at the bottom will internalize, but we focus on the cells that remain on the surface, which drive this elongation. And you can tell that the embryo is elongating because cells are flowing to the right out of the field of view as the tail is moving further away from the head. And what drives this elongation is groups of cells within this tissue are converging along the vertical axis and resolving in horizontal direction to the formation of these transient multicellular rosette structures where many cells come into contact at a single point. Now these behaviors are reiterated throughout the tissue and are associated with a doubling in the length of the body axis. Now, since our discovery of this rosette mechanism for tissue elongation, rosettes have now been shown to be a general mechanism for elongating many different types of tissues in vertebrates, including the chick and mouse neural plate, which are structures that have to elongate substantially to form the spinal cord. And these behaviors suggest that there's communication between cells. These cells are not just navigating on their own, but they're communicating with each other to form these groups that are able to rearrange more efficiently than the cells could achieve on their own. And we would now like to understand how these cells generate the forces that allow them to move and rearrange within the sheath, and how these movements are spatially organized so that each rearrangement inevitably moves the head further away from the tail. So to identify the molecular basis of these behaviors, we started by looking for molecules that are asymmetrically localized in the plane of the tissue and correlate with the direction of cell movement and could potentially be generating the forces that move these cells. And this slide shows the first two molecules that we found. So here you're looking at a field of around 30 cells, and in particular the myosin 2 motor protein shown here in green localizes to the surface the cortex of cells, but it's not uniform throughout the cortex. It's higher at these vertical edges between cells, creating the impression of vertical lines in this image. Now, the movement of myosin 2 motors along actin filaments generates contractile forces within the cell. And so this suggests that contractile forces that are planar polarized or oriented consistently within the plane of the tissue in this vertical direction could help drive cell rearrangement during elongation. HAR3 has the opposite pattern. It localizes to the horizontal edges of cells, where it stabilizes adhesion between cells, or the ability of cells to stick to each other, and it also excludes myosin, keeping myosin at those vertical edges. So through work from my lab and others, we've now identified more than 10 proteins that are include both signaling and structural proteins within cells that are required to organize these contractile and adhesive proteins within cells and are necessary for the embryo to elongate. So to look at the dynamics of how these proteins organize during elongation, Sergio Timulis in the lab made movies of embryos where myosin 2 is shown here in green and HAR3 is labeled in red, and time is indicated in minutes and seconds. And at the beginning of this movie, myosin 2 is mainly in the cytoplasm, so these circles you see are exclusion from the nucleus cells, and HAR3 is present in dots at the newly forming junctions between cells. And what you can see is that as the embryo gets ready to elongate, myosin 2 in green moves from the cytoplasm to the vertical edges of cells, and HAR3 in red becomes restricted to the horizontal edges. And what was striking about these movies is that myosin 2 is not just asymmetrically localized within cells, but it often forms these long extended cables that can connect multiple pairs of cells within the tissue. And this suggests that mechanical contractile forces aligned across many pairs of cells could generate the forces that drive rosette formation and cell rearrangement during elongation. So to test this idea, Rodrigo Fernandez Gonzalez in the lab used laser ablation to cut individual edges between cells in order to measure their mechanical properties. So in this first movie, Rodrigo used UV laser to cut a single interface between cells that's contracting on its own to drive local neighbor exchange. And you can tell that that edge was under tension because the two vertices attached to the cut edge move apart when that edge is ablated. And the speed and the distance of that retraction is proportional to the amount of tension that was acting on the edge right before you cut it, assuming that viscoelastic properties are uniform throughout the tissue. So this retraction distance is just over time, it's just plotted here in this chart. Now the movie on the right shows what happens when Rodrigo uses the laser to ablate a single edge, but one that's part of a multicellular cable that's going to contract to form a rosette. And what you can see is there's a much greater displacement of the two vertices attached to the cut edge as well as the surrounding cells. So these edges and cables are under more tension, and in fact, shown here in this plot, you can see that edges and cables are under twice as much tension as single vertical edges that contract on their own, and both types of vertical edges are under more tension than these horizontal edges in green, which have much less myosin. So these results tell us that mechanical forces are anisotropic within the tissue. They're highest in the vertical direction, and that they're and where they're highest in the sheath is in multicellular cables that are forming rosettes. And we've shown that these forces can travel up to tens of microns within the tissue and promote cell rearrangement and active elongation. So we now have a lot of information about the cell interactions that occur during active elongation, as well as the polarized force-generating machinery that actually drives these cell movements. But if we take a step back from thinking about what's going on inside the cell to looking at what's happening throughout the entire tissue, a major unanswered question is what orients these polarities across hundreds of cells so that they all move in the same way to promote tissue elongation. And it suggests that an additional system remains to be discovered that provides a global map that orients these polarities so that the cells all move in the same way to move the head further away from the tail. Now, the only clue that we had to the nature of this map came from a study by Ken Irvine and Eric Bischoff over 20 years ago, showing that stripe patterns of gene expression along the head-to-tail axis are necessary for elongation. So like us, flies have differences in cells along the head-to-tail axis, and in flies, these differences take the form of stripes because the body of the fly is segmented. In particular, even runs are two transcription factors that are expressed in 7 stripes along the head-to-tail axis, and this stripe expression pattern is critical for their function in elongation. So embryos fail to elongate normally when either Eve or run stripes are missing, or when either of these proteins is uniformly expressed throughout the embryo. So these proteins don't just have to be there, but they have to be present in stripes in order for the embryo to elongate. And we found that these stripes are not only necessary for elongation, but they're also sufficient to polarize cells. And the evidence for this came from experiments that I did with Eric Bischoff, where we generated mosaic embryos that have an ectopic source of Eve or run protein, shown here in blue. So, for example, in this mosaic embryo, a myosin marker is shown in green, and it localizes mainly to vertical edges of cells in the normal region of this embryo. But when you get to this clone of cells that has ectopic run protein, you can see that myosin polarity reorients to follow the clone boundary. And similarly, in this embryo that's mosaic has an ectopic source of Eve, you can see that myosin polarity in green reorients to follow the boundary of the Eve clone. So in each case, the orientation of cell polarity is dictated by the juxtaposition of cells that have different levels of Eve or run activity. So these results suggest a model where differences in gene expression along the head-tail axis of the embryo provides the spatial information that organizes cell movement during active elongation. But I mentioned that Eve and run are transcription factors, which means that their job is to localize to the nucleus of cells, bind to DNA, and regulate the expression of other genes. And so, because of their role in the nucleus, this model is ultimately unsatisfying. Because what we really want to know are what are the targets of Eve and run that localize to the surface of cells and allow cells to read the spatial cues in their environment that tell cells which way to move. So to identify these targets, Adam Perret, a postdoc in the lab, used RNA sequencing to compare gene expression profiles between embryos where both Eve and run were knocked down by RNA interference with water-injected controls. And he was looking for proteins that are targets of Eve and run that are expressed in stripes and that localize to the surface of cells where they could directly regulate cell polarity and behavior. So using this approach, Adam identified 3 transmembrane proteins in the toll receptor family as targets of Eve and run during active elongation: TOL2, TOL6, and TOL8. The toll was originally identified in Catherine Anderson's lab in Drosophila, and it's now known to represent a large family of proteins in both vertebrates and invertebrates. And these proteins have a conserved molecular structure, including a single transmembrane domain and a long series of extracellular leucine-rich repeats, as well as conserved sequences in the cytoplasmic domain. Now, these toll family receptors are perhaps best known for their role in regulating the innate immune response to pathogens such as bacteria, viruses, and parasites in the body. And in particular, this slide illustrates some of the phenotypes of mouse mutants that are lacking individual toll family receptors, and these mutants are defective for a subset of immune responses in animals. And the signaling pathway downstream of these toll-like receptors is very well characterized, with a major output of this pathway being the movement of an NF-kappa B transcription factor shown here in green from the cytoplasm to the nucleus of cells, where it activates the expression of genes involved in the inflammatory response. However, while some of the toll family receptors in Drosophila are required for immunity, the functions of most of the members of the family in Drosophila were not known. So since these genes are expressed in stripes, Adam first wanted to confirm that they really are regulated in some way by Eve and run, and that's shown in the images on this slide. So the top row of images show pictures of a wild-type or normal embryo, and you can see that TOL2 is expressed in many thin stripes along the head-to-tail body axis, and this pattern changes to become fewer, broader stripes in embryos that are missing Eve or run. Now, TOL6 and TOL8 are expressed in a slightly different pattern. They're expressed in several broader stripes, and this stripe expression is reduced in Eve mutants and is increased and is in a more uniform pattern in run mutants. Now, if you look at these toll receptors are expressed in a stripe pattern that requires Eve and run activity for their normal pattern, and you can tell that these 3 toll receptors are expressed in slightly different stripe patterns. And putting these patterns together, you get something that looks like a positional code, where nearly every cell expresses a different combination of 1, 2, or 3 receptors along the head-to-tail axis of the embryo. So the expression patterns of these 3 toll receptors relative to the segments of the animal are indicated here, and you can see that when you combine these 3 expression patterns, nearly every cell expresses a different type or concentration of toll these 3 receptors than their immediate neighbors to the head or to the tail. And the evidence for this type of positional code comes from types of experiments shown here on the bottom. So here Adam did a look at the mRNA expression patterns of TOL2 and TOL8, and you can see that the bright TOL2 stripes in red are largely complementary to the TOL8 stripes in green. And this image on the right is an image of TOL8 protein fused to a derivative of the green fluorescent protein, and you can see that it's also expressed from its endogenous regulatory sequences, and you can see that this TOL8 protein fusion is expressed in stripes and localizes to the membranes of cells. So these three toll family receptors meet our first two criteria. They're expressed in stripes downstream of even run, and they localize to the surface of cells where they could be directly involved in sensing spatial information in the environment that determines the direction of cell movement. So to ask if these proteins are required for elongation, Adam Furey used genome engineering to generate embryos that are defective for 1, 2, or 3 toll family receptors. And what he found is that although you get a reduction in elongation when you remove 2 receptors, you have to remove all 3 in order to see a strong effect. So working together with Zach Merman and Gene Farrell in the lab, they made movies of embryos that are missing 1, 2, or 3 receptors. And this slide shows a movie of a wild-type embryo on the top and an embryo that's completely lacking Toll 2, 6, and 8, shown here on the bottom. And time is indicated in minutes and seconds relative to the start of elongation. So this movie starts 10 minutes before the embryo is going to elongate. And the cells are color-coded based on the rearrangement that they start with. So they'll start off as purple, and then they turn blue as they go through one rearrangement, and then green, yellow, and red as they go through more and more rearrangements within the sheath. And if you focus on the top movie, you can see that the wild-type cells are very active. So the cells are turning colors. You see many green cells followed by yellow and then red cells appearing in the tissue. This is accompanied by elongation. So the cells are flowing to the right out of the view of the embryo doubling in length. In contrast, if you look at the bottom movie, the Toll 2, 6, 6 is much more sluggish. There's many more purple and blue cells remaining late in the process. And as a result, the elongation of the embryo is reduced by 30% in these Toll 2, 6, 8 triple mutants. So the toll receptors are required for cell movement, and fewer movements occur when they're missing. And when we follow the movements that still occur in these genes, we found that the cells that do arrange in this background are moving in a way that's less directional. So for cell rearrangements to produce elongation, the rearrangements have to occur directionally with respect to the head cell axis of the embryo. Otherwise, the cells would just be changing places, and you wouldn't get that elongation. So all of the rearrangements have to occur through the contraction of vertical edges separating these two cells that were initially in contact, and the formation of new horizontal edges between cells that were not previously in contact. And when we plotted the rate of errors in the second step of edge assembly, you can see that in wild-type embryos, these rearrangements occur directionally 90% of the time. In contrast, when we looked at the error rate of edge assembly in embryos missing 1 or 2 toll family receptors, what you can see is that the frequency of defects goes up to about 20%. And in embryos missing all 3 toll receptors, cells rearrange in a way that's not directional a third of the time. So, for example, in this schematic, a vertical edge will shrink to a point, and the new edge that forms is also staying the exact same edge as the edge that was there originally. So even though these cells went through the trouble of going through a cell rearrangement, no net elongation occurs as a result. And the defects in Toll triple mutants are similar to the defects in Eve and Runt mutants. So these defects in the orientation of cell movement are suggestive of a defect in cell polarity. And in fact, Adam found that these mutants have defects in the localization of the force-generating machinery within cells. So I mentioned earlier that TAR3 protein shown here in magenta is planar polarized. There's a difference between horizontal and vertical edges in the plane of this tissue, and you can see that there's more TAR3 at edges that are closer to horizontal and less TAR3 at vertical edges. And TAR3 acts in these horizontal domains to stabilize adhesion between cells and also to restrict myosin so that myosin is at vertical edges. And what you can see from this image is that in Toll triple mutants, TAR3 planar polarity is severely disrupted. TAR3 now localizes to edges of a variety of orientations, so you now see many more cells with TAR3, not just at horizontal edges, but at most or all the edges of that cell. And Adam sees a similar defect in the localization of myosin 2. So this tells us that these cells are not just moving in the wrong direction, but they actually seem to have completely lost their direction. They don't have the capacity to polarize the force-generating machinery in a way that would allow them to move in an organized way. So what I told you so far is that we have these 3 toll receptors that are required for cell polarity and for cell rearrangement during active elongation. And now if these are the key receptors that link the stripe information provided by the even Runt stripe transcription factors to the organized cell behaviors that establish tissue structure, then the key missing experiment is that these proteins should be sufficient to polarize cells, not just required for this process. So for example, I mentioned that expressing an ectopic source of E is sufficient to repolarize cells in the vicinity. And so we wanted to know, can an ectopic source of a toll receptor have the same effect if these receptors are the critical targets of even Runt during active elongation? Specifically, we wanted to know if in a field of cells that's not normally polarized, if you put in a stripe of a toll receptor protein, is that enough to polarize these cells? So to address this question, Athena Vicious, a graduate student in the lab, expressed the toll proteins in stripes in the late embryo, the stage when these cells are not strongly polarized. So you can see the control stripe, shown here in blue, does not induce a strong polarity of myosin 2 in green in this image, but in contrast, in embryos that express Toll 2 in the stripe, in magenta, you get a strong enrichment of myosin 2, specifically at the boundary where Athena induced a difference in Toll 2 expression levels. So this result tells us that the toll receptors are not only required for polarity, but they're sufficient to induce polarity when you introduce a local discontinuity or a stripe of polarity. So these are the key receptors that establish cell polarity in the embryo. This raises the question of what are the ligands that they bind to that allow cells to sense their orientation with respect to the overall axis of the animal. And in the immune system, these cells, the mammalian toll-like receptors, detect foreign molecules in the animal that are presented on invasive pathogens. But this is in the embryo in the absence of infection, and so there aren't predicted to be any pathogens present. And so we propose that maybe in this case, these receptors might be detecting receptors expressed on other cells. And for example, if the toll receptors interact homophilically with other receptors in the same stripe, then interactions between these receptors might recruit HAR3 to horizontal edges of cells. Alternatively, if these receptors interact heterophilically between stripes of cells that express different receptors, then interactions between the receptors might locally activate myosin and induce these cells to rearrange. Now, the stripe expression of these toll receptors suggests that they act as a positional code, and we're starting to decipher this code using studies in cultured cells. And these studies that Adam, working together with Chris Fincher in the lab, express the toll receptors in Drosophila S2 cells to ask, can these receptors